Hey, so today we're going to be talking about cell signaling. Um, we can start off with extracellular and intracellular signaling. So for extracellular, think of kind of like the extracellular matrix where it's on the outside. So extra for extracellular, you're going to think of on the outside, right? And so for extracellular signaling, you're going to think of signaling that occurs or starts in the very beginning outside the cell. So So when it comes to signaling, the molecule that you see right up there, right there, that's a ligand. That's usually the term we use for any signaling molecule. And then what you see right here is a receptor. As you can see, when it comes to extracellular signaling, the receptor is going to meet the signal on the outside. And the reason is because the signal is either usually polar, which means that it's not easily able to go into the cell, or it's huge and it's not, therefore it would require a lot of energy to push the membrane apart and bring the thing in. And so that's extracellular. Can you still see that?
Yeah, I got you.
Yeah. Okay.
Jump baby if you can.
Yeah.
Yeah, it's good.
Yeah. So when it comes to intracellular, the reason why it's different is because you're still dealing with the ligand, which is just a signal molecule. But in this case, for intracellular, most of the time it's going to be nonpolar. And so, because if you guys remember, if you guys notice in the membrane is made out of a very thin portion of polar heads, but then a very thick portion of nonpolar tails, right? So for a nonpolar molecule, although it's going to have difficulty crossing that nonpolarity or the polar heads that bind the membrane, it's not going to have difficulty going through that large section, and so it's easily able to pass through the membrane and into the cell without using a protein or using too much energy. And so, therefore, the receptor doesn't meet the cell or doesn't meet the ligand outside the cell, but rather it greets it when it's inside the cell because this is intracellular signaling. All right. Any questions about that? If you have any questions, make sure to maybe write a quick response on the comment sections. And if not, I'll just keep on talking about transduction. Yeah. Oh, but also, I have a question for you. What are some examples of ligand molecules that go with intracellular, which is this one, and signal molecules that go with extracellular, which is this one, that are greeted outside? You can post that in the comments, and I'll let you guys know if they're right. Don't be scared. Okay, so if you guys want, I can maybe give you some examples. So because we're working with polar and big molecules for extracellular, right? The reason why those for extracellular you need to greet them at the door right here is because they're too big or they're too polar to cross that big section of non-polar tails, and it's going to need too much energy to go through. And so, what are like, for example, sugars, right? Sugars. Right? For sugars, one, they're big, and two, they're polar, right? They have a lot of these polar hydroxyl groups all over the sugars, right? I think that's glucose. So they're big and they're polar, and so require a lot of energy to just go through or to go through the membrane. And so instead of wasting energy to push through these giant, big polar molecules, usually you just have the receptor on the outside of the cell, right? Therefore, extracellular. When it comes to intracellular.
Steroids.
So when it comes to intracellular, those are molecules. Usually the ligand is the one that I know I drew it kind of big, but usually it's very small or it's usually really nonpolar, right? And the example of O2, O2, O2 is nonpolar, and so it's easily able to go through the thing, and also it's a very small molecule. In addition, CO2, they're also very small, and if you remember, or if it helps you remember, CO2 and O2 usually those are gases, and so those are very small molecules that are able to go through. And the last example would be steroids. If the picture that I drew for steroids, it's a lot of like, it's a little big, but because it's so non-polar, it's able to go through pretty quickly. Yeah. For those that are signing in right now, those that are watching and you haven't signed in yet, if you can go to the description, there is a sign up or a sign in for CSS. Please do that. And also, yeah, like the chat says, if you have any questions, just type a quick question about this, or if you guys want to talk about something else about signaling, that's fine too, or anything else is fine. If not, I'll just keep on talking.
Or just talk about singular section. I guess, yeah, you should.
All right. So doesn't look like anybody has any questions. So we're going to continue and we're going to talk about just cell signaling in general. So if you want to take a picture of this, take it right now before I erase it. All right, sounds good. If anything, the live stream will be saved. And so you can just go on the channel and watch it again if you want. Okay. All right. So, what I drew is basically in general, what cell signaling is comes down to. So at first you have a molecule, right? In this case, does anyone know what kind of molecule this would be if it's being met at the outside of the cell? If you want, you could just quickly post it. If not, I'll just keep on going. So the first step, right? You have to have what you see right here. You have to have reception, right? So you have to meet the pizza guy at the door, accept the pizza. I guess in this case, pizza, right? Door. So you have to meet the pizza guy that's going to give you your pizza at the door, right? And so then, obviously, the pizza guy isn't going to come into your house. So you have to take the pizza and then take that signal and pass it on, right? But at the same time, you have this one big pizza, but you want to, at the second stage, you want to make sure everybody has enough. So you kind of multiply how much pizza you have. So that's what happens in the transduction stage, right here. Usually it's signified by arrows, but in those arrows is something we talked about in class when we have a kind of like amplification. So this one pizza molecule, right? Once it's recepted and it goes through transduction, it's basically like if for somehow you had a pizza machine that you can put a pizza in and it multiplied and started multiplying more and more and more and more. That's what happens in transduction. The cellular response is having a good time because now everybody has enough pizza to go around. But basically, in terms of the cell, usually this ligand molecule is recepted, and then it activates some proteins that we'll go through when we're talking about specific proteins like GCPR and stuff like that, or G proteins. And then eventually, once it amplifies enough, you have enough molecules to create a cellular response, whether that's creating more mRNA or making proteins or anything like that, just in response to certain things that are going around in the environment. But yeah, this is in general, this is what cell signaling is. And if anybody has any questions, post them on the chat. But I will go from here to a specific type of cell signaling that we covered in class, which is G proteins. So if anybody has any questions, make sure to post them. But I'm just going to keep them going. Oh, if you want to take a picture, go ahead. Mm-hmm. All right, so we're going to move on to G proteins. So let me just give me a second to erase this. Oh, okay. Is that correct? Or is it over here?
It's EI. I before E except after C and T in proteins.
All right. You can tell I'm not an English major. All right. So
I'm swells.
So let me just finish drawing.
Oh, nice.
Like a lima bean, right?
Yeah, and then
Yeah, that's good.
yeah, for now. You got other colors too.
I just feel like it won't show. All right, so G protein, usually it's a certain type of cell signaling, but it usually works under the same mechanism as extracellular that we covered in the very beginning of the live stream. But usually we start off with a certain ligand or signal molecule. That's what this is. It comes in and binds to this right here. Does anybody know what that might be? If you want, type it in the chat, but I'm just going to label it anyways. So this is so it's the G protein coupled receptor, and in the G protein case, it's not hard to remember the names of these things because two of them almost have the same name. One of them is the G protein coupled receptor because it recepts the signal. It's reception. That's the first stage that we covered right before this. The second stage is transduction. We'll get into that later. But what happens is you have this G protein, right? In the very beginning, before the cell signal binds to the GCPR, you have the G protein unactivated or inactivated. And so what that means is that it has GDP. And so basically, GDP is basically guanine, which, if you remember from the first exam, is a type of nucleotide. Nitrogenous base. Yeah. So nitrogenous base. So if you remember from the first lesson or from the first exam, so nitrogenous base, and it's bounded to two phosphates, right? Guanine diphosphate. So that's how you get the name GDP. All right. And so GDP doesn't have that much energy into it, but what does have a lot of energy is ATP and GTP. And the reason why is because when you have three different phosphate groups next to each other, that's a lot of negative charge really close to each other. And so they're repelling each other. And so it's very energetic and it's really unstable. And so when you have GTP attached to a molecule, or you have ATP and you use it as energy, the reason why you can use it as energy is because of that very unstable nature of the molecule. But when we begin, like I said, the G protein up here is inactivated because it has GDP. So the next thing that happens when this binds, this right here will the GCPR or the GPCR is going to change shape. And so when it changes shape, that causes a reaction to follow. And that reaction is that G protein is going to come here. And what's going to happen is it's going to replace this GDP. It's going to kick it out of the molecule. And then from inside the cell, right? Because this is inside the cell. This is inside the cell, right? Somewhere in the cell, it's going to find a GTP molecule that's just lying around, and it's going to take it up. Now, because it's taking away, or it's removing a GDP and taking in a GTP, it's gonna become activated now, right? Because like I said, GTP or guanine triphosphate is basically very energetic, and so when a molecule has GTP or ATP, it's considered activated because, in the term of this class, it's considered activated because of the energy that GTP holds within that molecule. So now I'm gonna change shape, and I'm gonna have now I'm going to have this G protein that's going to change shape because it's now activated, right? In general, when things become activated or when things are, yeah, when they become activated, they usually change shape. And then when they become inactivated, they also change shape. That's usually done with most proteins. That happened with G protein and then also with GCPR. That was a change in shape. So now this one, the G protein is activated, right? So now what it's going to do is it's going to go to adenyl cyclase. Going to go over here.
ATP. I'll erase that arrow.
What?
Yeah, you're good.
Okay.
Yeah, you're good.
Is that good?
Yeah.
Okay, cool.
I would remove the arrow from GTP to ATP because that like, makes it look like GTP is turning into ATP.
Is that better? GTP.
Yeah. Erase the triphosphate. Yeah.
So what I'm trying to show from here is that once the G protein gets to the adenyl cyclase, that enzyme is going to promote a certain type of reaction. And so what it's going to do is going to take this very energetic molecule of GTP. It's going to cleave it. It's going to break a phosphate from this GTP molecule, and it's going to create GDP, right? Because if you have triphosphate and you take one phosphate away, then you're left with diphosphate or two phosphates, right? So, but when you do that, like I said, GTP is a very energetic molecule. When you do that, you release energy. And that energy that you release, you can use that for different types of reactions. That's why UTP and ATP are used as energy currency of the cell. So what the energy is used for and what the adenyl cyclase does is that it takes that energy that's released and it converts ATP to CAMP. So if you guys are kind of confused on the abbreviations, CAMP is cyclic adenosine monophosphate. And so mono means one, right? And so you use that energy to go from adenosine triphosphate, ATP, right, to go to AMP, cyclic AMP, right? So you take off two, so that breaking off that phosphate is going to need energy. Even though it's very energetic, you need energy to break it, and then use that energy as well to twist it and make a cycle or a cyclic form, right? And that's what I kind of drew right here. Adenosine cyclic because it forms like a ring structure, and then with one phosphate. Adenosine monophosphate, but it's cyclic, so CAMP. So that's the purpose of the adenyl cyclase, all right? And then from there, we move on to transduction. But does anyone have any questions? Feel free to post them on the side or on the comments. And also, I don't know if we have any new viewers, but in the description, there is a sign-in for CSS like you usually do. Just make sure to like take a few seconds, go and sign in real quick, and then come back. All right. I'll give you like a minute to maybe think about this, digest it a little bit, and then if you have any questions, put it on there. I'll give you a minute. All right. So we're going to continue to transduction. Just give me a second so I can erase all this, but you take a picture. There you go.
Oh, we got a question.
Oh, you do?
Yes.
Okay.
Someone asked, what triggers the G protein to hydrolyze its GDP?
Yeah.
It's a good question.
Yeah.
Our first question.
Can you repeat the question?
So, what triggers the G protein to hydrolyze its GTP? No, she's corrected it to GTP.
Oh,
Yeah.
the first step.
I'm not sure.
So I'll just go over both. Just in
Yeah.
case. So that's a really good important distinction because in the change from GTP to GDP at the end is different than how it changes in the beginning, which I had up here. So yeah. So in the very beginning, right, when there's no signal molecule, you have GDP, which means it's inactive. So it's inactive, right? But then let's say the signal molecule binds, right? And so when it binds, it changes shape and tells the G protein right here, like, hey, get rid of your GDP and take up a GTP. So it becomes activated because when that signal molecule binds, it's like, hey, it's time to start the signaling process and all that stuff. So it's telling them, hey, kick out that GDP, get some GTP to become activated, and so we can continue this whole process. Right? It kicks it out. The next step, this one over here, this is different. So we have adenyl cyclase, right? That's an enzyme, and so it's going to promote this reaction where and it's going to promote the G protein is going to go from from over here where it's next to the GCPR, and it's going to go to the adenyl cyclase, and there it's going to come with the GTP, right? And so it's still activated, but it's going to bring it and it's going to present the GTP to the adenyl cyclase, and it's going to be like, hey, thanks, and it's going to cut off a phosphate, and it's going to how do I say this? The GTP is going to give the adenyl cyclase the GTP, right? And then it's going to be like, oh, cool, and take off a phosphate, and now you have GDP, and the G protein is like, oh, cool, thanks. And then I guess I'll just go back. And so it goes back to over here with GDP. So if the molecule is still there, if the molecule is still there, bounded, then the process is going to continue again, and it's going to tell it, hey, we're still connected or whatever. Let's kick out GDP again, take up another GTP, and then present it again, right? So in this step over here, when it's at the adenyl cyclase, it's going to be a cleaving of the GTP, not a kicking out of the GTP and taking up of the GDP, right? Because that makes sense because you want to create CAMP from ATP, and you're gonna need energy to twist the AMP to become a cycle to make a cyclic form. Does that answer your question, Monique? If that you still need if that doesn't, type something in the comments. But yeah, that's a good question because most students forget that it's two different processes. The first one is different than the last one. Okay, awesome. Yeah, that's a really good question, by the way. So yeah, I'm going to move on to transduction. If anybody has any other questions about this, feel free to interrupt me and post them on the comments.
B and C.
Oh, B and C? Yeah.
B and C, yeah.
Can you still read that? Is that longer? Yeah. Okay.
Yeah, you're good now.
Okay. So now we're on to the transduction stage. And so we just finished reception, right? Because we greeted the pizza man, we took the pizza in, right? And we continued the message, right? So now, after we convert that ATP to CAMP, that we're going to use that to continue the message, right? But before we go on, the thing we should point out is that for CAMP, that's considered a secondary messenger. That's what this abbreviation is, secondary messenger. What that means is that it's not directly the signal that caused this whole thing, right? The very first signal, the primary messenger, was that signal molecule that at the very beginning came and became bound to the GCPR, right? That was the primary messenger, right? But because it's not allowed to go in to the cell, well, we got to continue that signaling, and so we have to use a different signal inside the cell to continue it, right? Well, that's where secondary messenger comes in, and in this case, it's CAMP. Another example would be calcium, and when we're dealing with action potentials and stuff like that. That was on one of your worksheets. So this comes in and binds to a protein called protein kinase A. Protein kinase A, you can tell a lot from just the name. Protein, obviously it's a protein, and then kinase, right? Can you still read that?
Yeah.
Okay. So kinase, right? It's a weird word, but if you break it into pieces, the ACE is just a designation for any enzyme. Basically, any enzyme that you encounter is going to kind of have that ACE.
We have a question. Cyclic AMP is not a protein, correct?
Yeah, it's not a protein. So that was a good question. So do you see how I went from ATP to CAMP, right? The protein that was involved in that transaction was adenyl cyclase, right? Cyclase. Also, it also has an ACE because it's an enzyme. But yeah, you're right. ATP is not a protein. It's a nitrogenous base connected to 3 phosphate groups.
Energy molecule.
It's basically an energy molecule. And so is CAMP, right, in this case. Because CAMP is going to be bound to a protein kinase, and that's going to activate it. But before we go on, let me know if that answers your question, Jessica. But also, what I was going to point out was that for kinase, it's broken down into 2 things, right? We talked about what this ACE means. That means enzyme. Kin, usually, the way you can kind of think of it is, at least for me, because I'm a physics major, usually kin, I think of kinetic energy. And so a kinase would be an enzyme that gives kinetic energy, right? How does it do that? Well, in this case, what you don't see is that once this binds to here, it's going to give it a phosphate group. And so kinase is an enzyme that gives out phosphate groups to give kinetic energy to other molecules, and so therefore activating them. So if something has kinetic energy, right, like if I give you a phosphate and then you start like moving around and stuff, well, you have kinetic energy, and so you're activated, right? That's important to note because there's a different enzyme that does the opposite, and students get them confused all the time because they basically have they don't have the same name, but they're both dealing with phosphates, and so it gets kind of confusing. So yeah, like I said, protein kinase, right? Oh, it's important to know. So enzyme, right? I told you that the suffix for enzymes is ACE. For adenyl cyclase, right? There, it also has that suffix of ACE, and if you look, it's working with sil, right? Sil, as in like.
I think it's CL.
Is it?
Yeah.
CL.
CYCL. Cyclic.
Is it that? No.
Oh, you forgot another C.
Yeah.
Did I? Oh,
Cyclace.
cyclace.
And then before the L. CYCLA.
Oh my goodness. All right, so what I'm trying to point out is that cyclase, right? It's an enzyme, right? Because of the ACE that cyclizes, if that's a word, cyclizes a molecule. What molecule? Adenyl, which is the A in AMP and ATP. Does that make sense? Like honestly, if you are able to spell, unlike me, and you're able to break these words into their suffix and their prefixes and all that stuff, you honestly learn so much about what the purpose is. Like what it does is basically in the name, and that's what is cool about biology, I guess. So kinase is an enzyme. It's an enzyme that gives kinetic energy, so it activates it. So what, based on that description, we can assume that this protein kinase A is gonna it's gonna come to these other proteins, proteins B and C. It doesn't matter. The names aren't really important. It's gonna come to B and C, and it's gonna like tag them and be like, hey, you're gonna get a phosphate group now. And because it's a kinase, it's gonna say, hey, I'm gonna give you a phosphate group, and you're gonna become activated. And so that's what's going on here. All right. Now these two are gonna do the same purpose. They're gonna do the same function. They're gonna go to two friends.
E.
Thank you.
D prime. Get out of here.
Yeah. Okay. Okay. So it's going to do the same thing, but it's going to go to his B once it's activated. And the reason why I'm changing the shape of it is because when an enzyme usually becomes activated, like I said before, usually it's shown by a change in shape. So this B is activated, and so it's a square. It goes up to D, which is inactivated because it doesn't have a phosphate group. It gives it a phosphate group. It tells it, hey, I'm going to give you a phosphate group, and you're going to become activated. And that's what D prime over here is, right? It has a phosphate group, and now instead of being this oval shape, it changes into this square shape, signifying that it's activated, and also because it has, where is that? It has a phosphate group. And so it does that to both D and E. And so if we keep on going, right, I don't think I have enough whiteboard, but if we keep on going, you see that we started with one molecule being activated, and then the first time, now we have two. And now this one also will activate two other ones. So we started with one, and that one activated two more, and these two activated two more each, so that's four. And then once we go on, right, we're gonna activate a lot more. So by the end of it, we're gonna have like millions of like different proteins that are being activated. And so that's what we call transduction. Oh, let me write that down. Transduction. I need to move there. I think that's good. Okay, so that's what we call transduction. So basically, you start with one molecule, and you keep on pushing, or you keep on activating a bunch more, and so at the end you have millions and millions of molecules, right? And that makes sense because if you think about it, right, if you have one molecule in the very beginning, right, let's say you just activated this protein kinase and you went to go, let's say, get a cellular reaction or a cellular response, you have this one tiny molecule. And so it's going to have a hard time going into and finding the specific spot where you want to, let's say, do a specific protein. And two, it's only one. So like, how much power does that have to activate this huge thing, right? So you want to amplify the signal That way, there's more pressure to do a certain response or whatever. So this is transduction. Does anyone have any questions about that? Feel free to post on the comments. And yeah, I think that's mostly. Oh, just kidding, not done yet. So if let's say this is it, right? If this was it, and we had all these proteins activated, what happens when we are done and we get the response that we have, right? Like let's say we create a protein later on, and we created enough protein and we're done. Well, these can't stay activated forever, or else we're going to just use a bunch of energy creating this protein that we don't need anymore, right? And so something has to come by and take off these phosphate groups and deactivate them, right? Get them back to the resting state. So what that molecule is, or not molecule, enzyme. It's too far. So, I guess. So what that enzyme is called phosphatase. And so, like I said before, you see that ACE, think enzyme. The thing is, though, if you look at the rest of the word, it's phosphate, right? So you might be kind of confused if, like, let's say you're on the exam 3, you're on exam 3, and you're trying to remember which enzyme takes it away and which enzyme adds a phosphate group. Well, if you think phosphatase, you're like, oh, well, that one works with phosphates. And then you think kinase, you're like, well, there's no phosphate in the name. Oh, so it's obviously phosphatase is the one that adds the phosphate group because it's in the name, right? Which is, understandable. But that's why I made the distinction of, is it still on the board? Kinase, right here. Kinase is the enzyme that gives you kinetic energy. And so when you think of kinetic energy, think of like that P, as in like a lollipop or whatever that I'm giving you, and it gives you energy, it gives you sugar or whatever to make you more kinetic. Well, phosphatase does the opposite. And so that's why I made the clear distinction between kinase and phosphatase. But yeah, so phosphatase is going to come in. Phosphatase is going to come in. Sorry. It's going to cleave the phosphates from the enzymes, or sorry, not the enzymes, the proteins, C and B, right? And it's going to go from being square, being activated because it has that phosphate group attached, to being deactivated by taking off that phosphate. And so it goes back to its regular form. It goes back to resting, which is important because you don't want the signal to just keep on going and going and going and going, like I said before. So that's the role of phosphatase. That's the role of kinase, okay? And that's basically transduction. If anybody has any questions, make sure to post them on the comments down below. And if we have any new viewers, make sure to go to the description. There is a link to sign in for CSS like you usually do. So we can see, like, if you guys like it or not, and how many people signed in. Yeah, if you have any questions, I'll give you a minute to kind of digest this. I'm gonna move it a little bit. Yeah.
If you want to sub out.
And if you guys wanna have any questions and stuff, I'll give you a minute and post them in the comments.
you want to tap out, if you need.
It's really fun, actually. All right, so next we're going to move on from transduction into cellular response, which isn't covered in too much detail because there's usually a particular response that usually happens when we do this. So let me just give me a second so I can erase this real quick. And feel free to post any questions, even if I'm past this, or even about the very beginning of reception and G protein coupling.
Yeah, you can tell them it's like student led, like whatever you guys are
talking about. Yeah. Like Derek pointed out, this is kind of like, it's supposed to be kind of like a regular CSS. So if you guys have any questions at all, and really if it doesn't even have to do with this, just post it in the comments. Literally anything is fair game. We're just going over this right now because, you know, not everybody's commenting and all that stuff, so I don't want to just kind of sit here. So we're just going over this right now, but if you have any questions about anything else, literally anything, physics even, that'd be fine too. Just post it on the comments, and Derek will let me know if you have any questions.
Yeah. Dr. Latham commented.
Did she?
Yeah.
Hey, Dr. Latham. All right, so let's move on to cellular response.
I need some colors. I think which orange
Let's see how this looks.
That was red's bad.
Yeah, that's
Try purple.
Purple?
Yeah. Nope.
Oh yeah, I'm just gonna stick with black. Yeah.
Okay.
Is it muted? Okay. What is Halo?
He's doing almost CSS.
So, like I mentioned in the after, so this is after transduction, the thing that we just covered right now. And so we have a lot of activated, or we have this activated molecule right here that's going to go into the nucleus, right? This is. Did I spell it right there?
Yeah.
Okay. So it's going to go into the nucleus. What's in the nucleus? Well, that's DNA. So what we're going to see is that we're going to have this activated molecule. After we do transduction and all that stuff, it's going to try to get something to happen, right? Because that's the point of cell signaling, right? You have this outside source from the very beginning, right, that we talked about. We had this little tiny molecule called a ligand come in, bind, and say, hey, we need to create a certain type of protein or whatever. So it's going to pass that signal on through reception, and then it's going to amplify through transduction. And now we're at the end stage, cellular response, where we finally get that product that we're looking for. In this case, I'm just going to go through like protein synthesis. So basically we go to DNA, right? Well, what's the first step of protein synthesis, right? That's exam 2 material. So if you guys want to type it in the comments, let me know what you guys think. I'm going to keep going, but if you guys want to post it on there, that'd be cool. So this. We're going to work with a eukaryotic cell or eukaryotic cell. That's why I said RNA polymerase 2. But basically, this activated molecule is going to come in. It's going to bind to the promoter, yeah, and it's going to act as like a certain like transcription factor, right? Well, what do transcription factors do? Well, that's like we talked about in exam 2 stuff. It's basically going to tell this RNA polymerase, hey, come over here. We have this specific specific strand of DNA that I'm a promoter for, we need to transcribe it and then make a protein because we have this signal on the outside telling us that we need to make a certain type of protein or whatever. So it's going to act as a transcription factor. It's going to bind to the promoter. You guys all know this. It's exam 2 stuff. So it's going to do that. It's going to tell this RNA polymerase to bind, and you're going to create I'm including the step of splicing, RNA splicing and processing, right? But eventually, we all know that eventually you'll have an mRNA. Well, from an mRNA outside the cell, right? Because it's a eukaryote. So outside the cell, this is going to be used as a template to create the protein. Okay. Right. So that's a protein right there. So to summarize the whole step, right? We started with, after transduction, we started with an activated molecule, right? It's going to go into this nucleus, right? And it's going to bind right here. It's going to bind right here where it's binding at the promoter, right? Because once it binds to the promoter, well, that promoter is for a certain sequence of DNA, right? And once it binds, it's going to act as a transcription factor to try to get the attention of RNA polymerase 2 in this case, because we're working with eukaryotes. And it's going to say like, hey, come here. We have this strand of DNA that we want to transcribe and then make a protein out of. And so RNA is going to be like, okay. And he's going to go and he's going to bind, transcribe it. You create an mRNA after processing and splicing, obviously. Once you create that mRNA, you create that protein after translation. And so basically, like an example you can kind of think of is heat shock proteins. So the way that works, or what I'm trying to refer to, is that there's some types of what's it called, like cells that once, let's say you put them in a very, very, very hot environment, their first reaction, they'll have molecules that are excreted. I'm not particularly sure from where, but a molecule, a ligand, a signal molecule will come and be like outside a different cell, or outside you'll have a molecule bind to a receptor on the outside of the cell, tell it, hey, we're in a very hot environment. We need to create proteins or heat shock proteins that are going to help us survive in this very hot environment, right? And so this whole process goes, right? You get reception, then you go transduction, and then out of transduction, you get this. You have this process go on, and then you create that heat shock protein, right? Once you create that heat shock protein, it helps it survive in that kind of hot environment that it's not used to. But let's say we take that same cell that was creating all these heat shock proteins, and we move it to a regular environment. It's not going to need to make these heat shock proteins anymore, right? And so that signal molecule is going to detach, and the whole process is going to stop, right? Phosphatase to take off all those proteins, or the phosphates off the proteins that were activated in transduction. And then once that happens, we stop this from being activated. This last molecule that acts as transcription factor, we stop that from being activated. And therefore, we don't go through this process of transcription and translation. And that way we save energy, right? Like when that particular organism is in, or that cell is in a cold environment, or rather maybe just a normal environment, room temperature environment, then it's not going to need to create those proteins anymore. And so you don't want to waste energy doing that. And so that's how, that's like a kind of a real life example of what this, of this whole process.
Hey, David, I have a question.
Yeah.
It's not in the chat or anything, but how long, like relatively from like when the signal molecule initially binds, how long until we have a translated protein?
Yeah. Good question, Derek.
Yeah.
So it's important to know that, like I said, right? Let's say for that example of the heat shock protein, you put that organism into the heat into the heated environment, right? If this process wasn't done in like a very short time, it would be boiling alive, and it would take, like, let's say if it took an hour for this whole thing to occur, then it would get the proteins, but an hour later. But like, what if it's not alive then? So this process has to go very, very fast in a very short amount of time, which seems kind of weird because it's a huge process, but it's biology and everything goes pretty fast, especially when it comes to life and death situations like the heat shock proteins. Yeah, if you have any other questions, post them on the comments. From here, we can go into other types of signaling. We can go into tyrosine kinase receptors, which are very similar to the whole process, but it's only the reception that changes. We can go into that. Well, that's what I'm going to go into. But if you guys have any questions about this, or transduction, the step that we did before, or even reception of G protein, post them on the comments and I'll make sure to get them. Even if we're way past that, doesn't matter. It's whatever you guys want to talk about. And also, if I don't know if there's any new viewers, but there is a sign-in, kind of like for a regular CSS. So in the description. So go to the description, click on the link and sign in. Just so we know how many people came in and watched it, even just for a few seconds. But yeah, I'm going to go into tyrosine kinase receptors next. Just give me 1 second to erase all this. Or if you guys want to take a picture.
All right. You can talk about. Can you talk about tyrosine?
Okay, so we're going to move on to tyrosine kinase receptors. If anybody logged in, we just went over cell signaling from the very beginning, or the very beginning, from reception to transduction to reception, or sorry, to cellular response. That's what's on the board right now, and we covered it in general as well. If you have any questions about that at all, put them in the comments. It's whatever you guys want to talk about, but I'm going to keep on going into tyrosine kinase receptors if you guys want to just follow along. Just give me a second so I can erase the board.
Sitting back to me,
If I move it back over here.
yeah. Just move it. Just move it
All right,
back
that's
to
better.
what you were. Remember?
Okay.
We don't have it. We have till 12.
If you have any questions while I'm drawing, put them in the chat. That way it's not so awkwardly silent in here. All right. Okay. Yeah. So, like I said, I kind of mentioned it before. When we're talking about cell signaling, the thing that changes a lot is, okay, thanks. The thing that changes a lot is the reception, right? So we talked about one. We already went over G protein and G protein coupled receptors. That's one type of reception. The next or the other type of reception is tyrosine kinase receptors. Those are kind of look like this. And so what happens is the biggest difference is that you need two signal molecules to activate this. So just to recap, because we're doing cell signaling and this is extracellular, and these ligands or signal molecules, they're either polar or big, and that's why you have to have these protein receptors meet them outside the cell, right? Because if these were nonpolar, they would just be able to go through, or if they were very small, they would be able to go through this membrane here. If you guys have any questions about that, post them in the chat. I'm not able to see who's been here, so I'm not sure if like there's new people that weren't here from the very beginning. So we talked about that, but if you have any questions about that or you want me to go over that really quickly, just put it in the chat. So these ligand molecules, they're going to bind to these protein receptors, right? In this case, they're tyrosine kinase receptors. So the reason the way this works is you need two signal molecules to bind, right? The reason why is because these need to come together. And so once they do bind, which is what I'll draw. Once they are binded, this receptor is activated, right? And so if you guys remember from the G protein example, once the ligand molecule was bound to the G protein coupled receptor, it changed shape, right? It changed shape. In this case, it also does the same thing. It changed the shape, right? It went from being separate to being a dimer, right? So as long as those molecules up there, those signal molecules are attached, it's going to be a dimer, it's going to be one thing, and it's going to be activated. So just give me a quick second. Yeah, so I was just making sure I had the right idea. So for tyrosine kinase, all right, we kind of pointed out, like we said before, this ACE stands for enzyme, right? That suffix stands for enzyme. Kind of similar to when we saw the protein kinase, that stands for kinetic, or not stands for, but you can kind of remember what it does by remember that ACE represents proteins, and kin, you can imagine that it's a protein that gives kinetic energy. And so you're giving kinetic energy. Well, how do you do that? You add a phosphate group. And so that's going to come into later, but also the name tyrosine, right? Well, we haven't drawn anything that's related to it, so you might be wondering where that name comes from, which is a good question. So on the sides right here, something so the tyrosine, the part of the name or the name tyrosine kinase receptor comes from the tyrosines on the bumps of this protein right here. And so what happens is that you have, for this receptor to continue the process, these tyrosines, right? Which, if you guys remember, tyrosine is an amino acid, right? These tyrosines are going to be phosphorylated. Right. So basically, these bumps on the outside are going to be phosphorylated, and so what that does is it's able to, or sorry, once those ligand molecules bind to those proteins, they come together, and then they have these tyrosines on the bumps of these proteins that will become phosphorylated. Okay, what that does, and what the purpose of that is that you have yeah. So what that does is that the same thing happens on the other side. But you have these other proteins, right, that are going to be attracted to it because it has those phosphate groups, and it's going to come to it, and then that receptor is going to phosphorylate these proteins right here that are inactivated right now. But once they come to the tyrosine receptor, they're going to become phosphorylated themselves, and those are going to become activated. Okay, and that's going to happen for each one that comes to the receptor and becomes phosphorylated. And the same thing happens on this side. I'm just not going to draw it twice, but it's the same thing. And that's going to keep on going until these molecules, or as long as these molecules are bound, right? As soon as they come off, then these phosphate groups are going to go away, and you're gonna see the change, or you're going to see a change in shape again. But this time it's going to go back to its resting state where it's separated. Okay, and so if anybody has any questions about that, put them in the chat or in the comments. And
David, I have a quick question.
no, it's up there.
So how do those tyrosines become phosphorylated?
Okay, good point. All right, so I forgot to mention that how these become phosphorylated, right? It's a term called autophosphorylation, and so they phosphorylate themselves. And so once they become phosphorylated, they become activated, and it's able to, at one time, activate 6 other proteins, and 3 other over here. I'm just gonna draw it. And so it's able to activate 6 other proteins, right? And so you could see that it's very different to G-coupled protein receptors because there was that the initial kicking out of GDP to take in a GTP, right? And then it had to go across the membrane to adenocyclase and then create a secondary messenger and all that stuff. And that's still a part of the process when it comes to tyrosine. But the very beginning of this, the very beginning or the reception part is going to be very different because for this one, there's autophosphorylation and there's also that beginning change in shape where it comes together. But after this, once we have these proteins that are activated, it's going to follow the same type of pattern of transduction and cellular response that we saw with, or that we went through with G proteins. So really, the only thing that changes, like I said, is the reception. And that's it. Any questions about that? Make sure to put them in the comments. And also, if there's anyone that's new, make sure to go to the description. There is a link to sign in for CSS, just like any other CSS that you go to, hopefully. And just sign in and just come back, I guess. But yeah, just going to give you a quick minute if you guys want to digest it and then maybe post a question, or if you want to take a picture or screenshot.
take it over.
I will be right back. Jorge, your lovely CA, is going to sub in for me for real quick. Just an announcement that we're going to finish pretty soon. So if you guys have any questions, Jorge is going to go over some other material as well. But if you have any questions at all before we end the live stream, we'll give you guys a heads up. But if you have any questions, make sure to post them.
How many people are there?
Just kidding.
Okay, guys, so we'll be finishing in about 7 minutes. If anyone has a question, feel free to ask now. Just give you a couple seconds. See if you,
louder, buddy.
you think they can't hear me?
I don't know. It just might be easier.
Okay.
Let's see.
Does anyone have a question?
They're not even probably. Yeah, actually, I can just talk about it.
Okay. So I guess if no one has a question, nobody has asked anything yet. Okay.
I'll keep watching.
Okay.
I got you.
Where's the eraser? Do you have it?
He knocked on the ground. Oh, there is a question. So Jessica asks, this is a complete different topic, but self-signaling insurance section. But can you explain the generation of an action potential?
Okay.
It's just very different. But yeah.
Yeah. You think she's asking about like the what do you call the
But like threshold?
This is
Yeah.
good for you to use some kind of questioning, so you can ask her to clarify
let's
by
see.
asking another.
do you want to do that?
Yeah,
Ask
yeah.
on the chat. What do you think she?
I just asked.
Okay.
Explain the generation of action potential. I guess you can just draw your neuron.
Wait. So what does she ask? I don't see it here.
You don't see it?
Mm-mm.
Can you explain how it travels
There it
down?
is.
How an action potential travels down the neuron?
So just how would you say that? Just like synapse or?
Yeah.
Is she just
Like go from initial depolarization to
have they gone over the graph yet? We can go. over that.
yeah.
Maybe she should be that like asking about like.
I got it. Yeah.
Well, I don't know if you guys remember this from class, but we call the. You hear me? Okay.
Yeah.
From class, we call this the. Well, it's just like this like area right here, the synapse, like the in between.
Don't talk to the board.
Oh, sorry. Yeah. So basically, what's happening is there's neurotransmitters coming from here, from up here, and they're released in the synapse, and there are receptors on the surface of the target. I think that's what she's asking, right? Or
How it travels down the neuron.
yeah, okay. So it's just your neurons, and this one just releases the neurotransmitters, and this are, like I said, just releasing the synapse, and this synaptic cleft, which is a better word. That's what it's called, right? The synaptic cleft.
Yeah. It's the same word.
Yeah, it's the same thing. And just remember that the next, I guess the target where it's going, there's receptors on the surface, and the neurotransmitters bind to the surface, to this receptors and this is gonna trigger a response. It can either be like there's it's just something coupled to this receptors, and then I think you guys went over this in discussion, but the neurotransmitter doesn't go through here. This only releases a response inside the next.
You're good.
Here. Let's see something.
No, you're good. There's a lot.
I think she's asking about
How it propagates down an axon, not.
so basically action you know the graph.
Yeah,
I think that's what she's asking about and how
like
that
just
would-
go for the graph or.
yeah, and talk about how like that's propagated. Like basically remember how it does it and then
Like
the
opening
next
the
part of
NA
membrane.
the sodium channels and all that, or
Yeah, and then the next part of the what is it called? The not the yeah, the axon. And how it propagates down the axon. So like there's
Yeah.
an action potential here and then the next one's at the end of the other one's at the end.
Or do you want to help me with like something really?
Sure, yeah.
You got it.
Not really sure here.
Yeah, because you're talking about how she's asking down a singular neuron. You were talking about how it jumps from neuron to neuron.
Okay.
Both are important.
So basically, when it comes to Jessica's question about how it travels down the neuron, you guys all know this graph. Right? And so you all are pretty familiar with that graph, right? It's the action potential here, AP, and this is the potential
Voltage over time.
time, right? Okay. Yeah. So this is voltage, membrane voltage, measured in volts. So what you have is you have this process, right? And you guys went over this in class, where you have, sodium channels open up, you have potassium channels open up, and you create this action potential, or you create this change in membrane potential. And once you change it enough, you have this very high rise in membrane potential that you reach this top point where you get an action potential, right? So what this looks like is like kind of
Yeah.
So then the dendrite is
That's your cell. You need cell body.
the cell body is like
Yeah.
okay, that's overlapping. But what I'm trying to show is that this graph right here, this change in action potential, is occurring right here, right? Then once that action potential is done here, that this membrane is going to change, it's going to create the action potential, and then the next part of the membrane right here is going to go through the same process again to travel. It's going to open up the sodium channels, it's going to open up the potassium channels. That way it creates its own action potential and therefore kind of propagates it down this area right here of usually a neuron. It propagates it down this area of a neuron. If anybody knows what this is called, put it in the comments. It's called an axon, But if you knew that already, put it in the comments. And so it then after this does basically like that. Yeah, and then so basically it does the action potential here, right? It goes through the membrane potential here, the change in, and then the next part does it as well. That way it kind of continues the message, and then the next part of the axon does it, and then the next part and the next part until it gets to here, right? Until it gets to the cleft, right? In this space, let me move it a little bit. This space right here, this empty space, that was what Jorge was talking about before we talked about this, where you have the neurotransmitters leave and you have them go on to the next cell, right? Oh, no. Yeah. It would leave this one axon, or sorry, it would leave this one neuron and then travel to the next neuron. right? And then the same, like let's say this keeps on going, right? Then the same thing would happen to this one. It would receive the message, and then it would also do this again, where it propagates it down and to the next neuron. And so it just keeps on going until what Jorge was showing, which was when the neuron connects to a muscle cell, and then it creates an action like contracting or something like that. Yeah. Does that answer your question, Jessica? Put it in the comments if it does. But yeah, that's how it propagates it. It just does its own action potential across the membrane. And that's how you get from receiving a signal here to, awesome, yeah. So receiving a signal at the end, at this cell body, right, and the dendrites, to getting it to the end of the cell and then pushing it on to the next cell, right? Because if it stayed within this one cell, well, then you wouldn't be able to pass along the message and it would just die there. So you want to make sure you pass it along these axon, and then at the end, pass it on to the next cell. So, I'll just give you a minute to kind of digest that and post comments or questions on the comments.
Or is there a question you can
Hey guys,
ask?
just so you guys know, if there's a new viewer, there's a link in the description to sign in, even though it's like almost done. But we're almost done, and so if you guys have any questions, oh yeah, if you guys have any questions before we end the live stream, post them. We'll go through them right now. And also, if you are barely joining us at the very end and you missed kind of like the very first 30 minutes or the first hour, this is going to be saved and it's going to be posted on this channel. So make sure to like and subscribe and go in the comments and be like, oh, I hated David's voice, or hey, Jorge is amazing, or anything like that. We'll take any criticism. And also, just if you guys want to, if you feel the need to, go to the comments, tell us if you liked it. And yeah, so I'll give you guys a few minutes to post any questions. And if there's no more questions, we'll end the live stream, and then we'll shortly post the whole video. We'll post it in the next coming days to the channel. And so if you want to just review the whole thing, or if you want to just kind of listen to it as a podcast or whatever you guys want, we'll give you like 2 minutes, and then to post some questions. So that those that just joined, we're just taking questions for like the next minute and a half. We're gonna end the live stream pretty soon. But if you have any pending questions about anything, ask them in the comments, and we'll try to get to them before we stop. But then if nobody has any comments or any questions in the next minute, we'll end the live stream. And if you haven't signed in yet, please go to the description, click the link, and sign in just like any other CSS, and also comment, like, subscribe the video, everything.
Turn on notifications.
Turn on notifications so you guys know when we post these. And also, yeah, so 1 more minute and then we'll go offline. So it doesn't seem like anybody has any last minute questions. So we're gonna end the live stream. Thanks again for watching. If you enjoyed it, Derek will be in class tomorrow. So tell him all about it. Tell him if you loved it or hated it. This is a new thing that we're trying to do. So any feedback at all would be very useful. And yeah, that's about it. Make sure to subscribe so you can hit the bell icon so you get notifications every Thursday, if this keeps on going, that we're live streaming. But yeah, bye guys. Oh, are you sure? Yes.